Ever wondered how scientists decode the complex language of life hidden in our DNA and RNA? Welcome to the captivating world of nucleic acid sequencing technologies, the powerhouses that have revolutionized the field of genomics and bioinformatics. The journey of these technologies has been nothing short of remarkable, starting with Sanger sequencing. This method, named after its developer Frederick Sanger, was a breakthrough in its time, allowing us to read the genetic code for the first time ever. But science didn't stop there. The quest for more efficient and faster ways to unlock the secrets of DNA led to the development of next-generation sequencing, or NGS. This technology took us from reading single DNA fragments to millions of fragments simultaneously, a quantum leap in our ability to understand the blueprint of life. These technologies have dramatically reshaped our understanding of biology. While these technologies are impressive, they each come with their own strengths and limitations. Why does one scientist prefer using Sanger sequencing while another chooses next-generation sequencing? Well, let's dive into it. When we talk about Sanger sequencing, we're referring to a tried-and-true method. It's reliable, particularly when dealing with small genome sequencing. But here's the catch. It's not quite as efficient when it comes to larger genomes. It's a bit like trying to read an entire encyclopedia one sentence at a time. Doable, but not very time-savvy. Now let's turn to next-generation sequencing, or NGS. This is the high-speed train of the sequencing world. It provides high-throughput sequencing, getting you that genetic information quicker. However, as with all things, it has its limitations. The main one being short read lengths. It's akin to trying to piece together a puzzle without being able to see the full picture. Choosing the right sequencing technology depends on the demands of the research question at hand. And that's the crux of it. How do scientists make sense of the enormous data generated by these sequencing technologies? They use the power of bioinformatics, a field that combines biology, computer science, and statistics. Bioinformatics tools play a vital role in analyzing and interpreting sequencing data. Let's imagine the process. The tools take raw sequencing reads and align them to a reference genome. This alignment forms the basis for variant calling, where the tools identify differences between the sequenced DNA and the reference. They then assemble these variants into a complete genome, and finally carry out functional annotation to understand what each gene does. However, these tools aren't without their limitations. They require a level of computational expertise and the ability to handle big data, which can be a challenge. Also, the sheer volume of data generated can lead to errors in interpretation. Bioinformatics tools are the unsung heroes in the world of genomics, turning raw data into meaningful biological insights. So, what's the takeaway from our exploration of nucleic acid sequencing technologies and bioinformatics tools? It's clear that while these technologies and tools each have their unique strengths and limitations, they're crucial in propelling our understanding of life's genetic framework. They help us unravel the complex mysteries of the genetic code, laying the foundation for numerous scientific breakthroughs. Next time you hear about a breakthrough in genomics, remember the intricate dance between technology and biology that made it possible.